Uh, Hello. I would like to begin by acknowledging the Wurundjeri people of the Coli Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today. I pay my respect to their elders, past, present, and emerging, and to the Aboriginal elders of other communities. I would like to introduce myself first. I'm Mayada Mazhar, a center PhD student at Uni Melbourne. And as a part of housekeeping rules for the seminar to run smoothly, dear audience, you should stay muted throughout the presentation. Kindly leave all questions to the end of the seminar in the Q&A session. In case of you have questions during the seminar, send it in the Zoom chat and it will be read out later in the Q&A session. The seminar will be recorded and the recording will be shared on the center website. I'm happy today to welcome Dr. Alastair Stewart. Alastair is a laboratory head at the Chang Cardiac Research Institute in Sydney, Australia. Throughout his career, he has used a complementary spectrum of structural and biochemical methods to unravel the function of protein complexes important in biomedicine. Alastair embarked on his scientific journey at the University of Cambridge, where he read the natural sciences triples. He subsequently moved to Sydney to pursue a PhD focused on deciphering the mechanism of ATP synthase. Following his doctoral studies, Alastair undertook a brief postdoctoral fellowship at the University of Sydney, delving into the regulation of chromatin by long non-coding RNAs. Before setting up his independent research group at the Chang Cardiac Research Institute in 2016, Alastair has been the recipient of 3 consecutive fellowships from the National Health and Medical Research Council, most recently being awarded the Commonwealth Minister's Award for Excellence in Health and Medical Research for highest ranked emerging leader investigator granted in 2023. Today's seminar is entitled Transporting Drugs and Protons, E. coli, ATP synthase, and human OCT1. Hello, Alastair, and the stage is yours.
Thank you very much for that kind introduction, and thank you very much for the invitation to speak here at this CCEMP research kind of symposium series. So as you said, I'll be talking today about transporting drugs and protons, looking at a couple of different protein complexes, both F1Fo ATP synthase and also OCT1. But first of all, I'd just like to take an opportunity to thank and acknowledge the people that I've been able to work with on these projects over about the last decade now, really, although really ramping up over the last 5 years. First of all, my group, this is a bit of an old photograph now. Some of you may know James Walsh, who's moved to the Max Planck in Göttingen to work with Patrick Kramer, and also Emily Furlong, who's part of this center, who set up her own independent research group at ANU. But the work I'll be talking about today was mainly driven by both Jack Zheng and Meghna Sopti. Meghna's been working on ATP synthase, and we've been lucky enough to work with quite a few collaborators in this space, particularly Hiroki Noji and Boshi Uno, who were the world leaders on doing single molecule science on F1 ATPase, so looking at rotation of F1 ATPase. But we've also been lucky to be able to work with Richard Berry and Robert Ishikahamatov, who work on the F1Fo ATP synthase, which contains its membrane domain as well, at the University of Oxford, who also do single molecule research on that complex, and Carol Robinson, who does intact mass spectrometry for us on those complexes. Jack's been driving work on this human organic cation transporter 1. He's a PhD student co-supervised by myself and also Jamie Vandenberg and Renee Ryan, who's spoken in this series as well. And together, they've been helping really mentor looking at this complex, both from a structural and functional point of view with assays, but we've also collaborated with Megan Amar and Ada Quinn at UQ to do molecular dynamics, Nicholas Smith at UNSW to look at activity assays. But as we're mainly structural work, we wouldn't be able to do any of this work without a fair bit of mentorship, also in the microscopy, in order to do cryo-electron microscopy. It started with Sarah Sandin with a very nice collaboration when she was in Singapore at NTU. And she taught us how to make grids, screen them, really understand microscopy and begin to process data. And this was driven further by Craig over in Portland at OHSU, who really showed us to take, I suppose, high-end data and different ways of processing the data. And more recently, as part of the CCEMP, through James Bauer and Simon Brown, who were at the University of Wollongong. And most of this work has been funded by the National Health and Medical Research Council through project grants and fellowships. So as well as the people and the organizations, I think it's important to acknowledge the instruments we use as well. We were lucky enough back in 2016 to get a grant from the New South Wales government to buy a Thales Arctica. And we installed that in partnership with the University of New South Wales, Sydney, who were able to screen grids there and certainly take data. And then after that, we usually take our data to a couple of facilities, most recently Molecular Horizons down at the University of Wollongong, again run by James Bauer here as part of the CCMP, and also over in the States in Portland at the PNCC, Pacific Northwest Cryo-EM Center, which was partly funded by the NIH. And by using these instruments about 2018, since we installed our Thales Arctica, we've been able to look at a range of different complexes. On the left here, this is mainly the work from my group, working on ATP synthase, quite a large rotary motor, which is membrane bound. It has a large soluble domain I'll talk about today, and also a membrane domain that we're trying to understand its function. Also these small SLC22 proteins, which are only about 60 kilodaltons, which have mainly membrane bound, but also have quite a large extracellular loop that we can use for orientation. These very large chaperoning complexes, which have high symmetry and enormous, that we were interested in how they're able to form different complexes made up of different subunits. As many people, we looked at the SARS-CoV-2 spike protein, that we had to chop its membrane domain off like everyone else, but bound to different types of therapeutics, both Fabs and other experimental therapeutics. And then collaboration, we've also looked at the Herd potassium channel, about a 0.5 megadalton C4 protein. And although this work is mainly run by Jamie Vandenberg, I guess we facilitated them using the microscope and hopefully mentored them in that. Renee Ryan looking at glutamate-like transporter, GLTPH, able to get us set up with cryo-EM data, which they're able to, she's presented this work as part of the series. Hopefully you've seen that before. And then more recently working with Joel Mackay over the University of Sydney as well, looking at the nucleosome core particle, taking very high resolution, making very high resolution reconstructions of that, and also trying to get it bound with different things. So you can see mostly we've worked on proteins which are inside the membrane. So hopefully people at this seminar will be interested to hear about those. And today I'm just going to be talking about these two complexes here, a complex ATP synthase. First of all, looking at just the soluble part, F1 ATPase, and also the whole complex F1Fo. Then moving on to work that my PhD student Jack's been working towards and understanding the cellular carrier 22 family. So to begin with ATP synthase. So hopefully anyone who studied undergraduate biology or at least high school biology has heard of this molecule ATP, often referred to as the energy currency of the cell. And it's called ATP because it has 3 of these phosphates, adenosine triphosphate. And this molecule can be hydrolyzed into ADP and phosphate. And when it does so, because these products have a lower delta G, significantly lower delta G versus the reactants, this is an energetically favorable reaction. And it gives, I suppose it gives out about 60 kilojoules per mole in delta G. And biology is quite clever. It's able to use this energetically favorable reaction and couple it to other unfavorable reactions, energetically unfavorable reactions. These might be conformational changes that are involved in polymerization, transport, or a myriad of other biological processes. But in order to use this, biology has to recycle this. It has to synthesize that ATP back from ADP and phosphate. And the majority of this in most cells in biology is generated by this molecule here termed F1Fo ATP synthase. So you can see on the top left a schematic. This might be a mitochondria or a bacteria in this case. And you have a series of complexes which are able to use factors in order to pump protons from one side of the membrane to the other. And when they do so, they build up this proton motor force, and there's this proton motor force which drives protons from that membrane back, in this case, into the cytoplasm of a bacteria. And this is used to cause rotation, which in turn generates ATP from ADP and phosphate. So about 10 years ago, we made a movie to describe that, which is a bit out of date now, but hopefully gives an overall depiction of this. And hopefully at the bottom, you can see in this video, we have these highly energetic protons, which are on one side of the membrane. These bind to this rotor subunit in this membrane-bound FO motor, causing rotation of the central stalk. And this in turn causes conformational changes in this group up the top here, which is where it provides that catalytic environment to synthesize ATP from ADP and phosphate. Now, the top piece here, which is soluble, we refer to as factor 1, or sorry, F1, and that's just because inhibitory factor 1 binds here, so we call this F1. And at the bottom, we call this FO because oligomycin binds at the bottom. And what's key here is at the bottom, you have this membrane electrically powered, effectively motor, which is generating this rotation. And at the top here, you have this F1 motor, which is using that rotation in order to catalyze that reaction. So it's a chemical motor up the top. So we're certainly not the first to solve structures these complex or work in this field at all. And I've just shown some examples here by work by John Rubinstein, John Walker, and Werner Kubrand when they were working on mitochondrial F1Fo ATP synthase. You can see here as dimers, ours is just a monomer. But I guess there were many things to learn about this whole system, partly how they look like in mitochondria, how they function, and what we can learn from that. But what my lab's focused on is mainly these bacterial ATP synthases. And we've done that for two reasons. The first one is they're antibiotic targets. So antimicrobial discovery is certainly interested in these complexes in general. But we're also, from a fundamental biology point of view, these complexes have been known for sort of 20 to 30 years. So there's a vast array of mutagenesis data and also single molecule data that we're interested to see that if we can link this up to structures where we can really understand the function of how this complex works. So to begin with, again, I said I'm just going to be talking about this F1 ATPase and the work being done to try and understand how this chemical motor works. And then I'll show you some other work that we've done on the whole complex, this F1Fo ATP synthase solubilized in detergent, to try and understand how the membrane-powered motor works and also the how whole enzyme generates ATP. So again, if you've done undergraduate biology, this is something that I briefly went over in my first year biology of cells. You might have heard of this binding change mechanism of the F1 ATPase, and this was an idea that was pushed by Paul Boyer in the 80s and early 90s. And what he essentially said was that at the time they knew there were 3 binding sites on the F1 ATPase, the soluble piece at the top, and a gamma subunit in between. So he's shown this as schematic. And if we're in synthesis mode, so we're driving this with protons, this would cause rotation of this gamma subunit here. And if you look at these 3 binding sites, you might have one where ADP comes and binds and phosphate binds, so it bind well here. You then have a rotation, and this will provide the environment in order to catalyze and generate that ATP. It then rotate again 120 degrees to an open state to get release of ATP, rotate round again, bind ADP, and by cyclically stepping 120 degrees, you'll be able to drive this synthesis of ATP from ADP and phosphate. Now, with most enzymes or all enzymes in chemistry, we have this irreversible reaction. So you could actually, rather than synthesis, you could also have it in hydrolysis, which is often how we look at this. And in this sense, instead, you'd have the F1 ATPase would be hydrolyzing ATP, be driving rotation. So here, ATP we had to bind. This would cause some form of rotation. You get to some block where you'd have hydrolysis, some gate, and then this would cause rotation again, and you'd get release of your products, ADP and phosphate, and you'd go around again. And this idea was all but confirmed by a crystal structure that was generated by Johan Peter Abrahams and Andrew Leslie and John Walker's group. And what they're able to see here is this 3-fold symmetric complex with this gamma subunit in the middle here. And usually they've shown here is yellow and red subunits. These are called alpha and beta subunits. And generally in the field, we effectively ignore the alpha subunits, these red ones, because although they bind nucleotide, they usually bind ATP, they're non-catalytically active, so we don't talk about them. So instead, I just went to concentrate on these yellow subunits, and you can see they've labeled one empty or open, the beta empty, and this doesn't have nucleotide bound. They had another one which had ADP bound and was closed, and another one that had ATP bound and was closed, again confirming this idea of a binding change mechanism. We'd have empty, ADP bound, and ATP. So another beautiful piece of work was the beginning of single molecule studies where Hiroki Noji in the lab Yoshida and Kinoshita was able to, for the first time, visualize physical rotation of this complex. So here they took, again, this alpha and beta, this yellow and red subunits, and the gamma subunits, and they put a very large fluorescent actin filament on there. And when they observed that through a fluorescence microscope, they saw rotation. And more than that rotation, they could actually see that it dwelled in these 3 different positions because it was getting catalytically stuck. It catalytically dwelled in those positions. So as well as seeing rotation, they could even see this 120-degree stepping rotation as that fluorescent actin filament moved around. They took this work quite a bit further a few years later, where they replaced that enormous actin filament with a very small bead, and they changed the way that they were able to image it. And by doing so, they could get much higher temporal resolution of this. So they could see these objects on sort of thousands of frames per second of scale, up to 100,000 frames per second scale. And here you can just see the centroid of this beam in this view, hopefully you can see is just rotating around. And the astonishing thing they saw here is rather than having 120-degree steps, when they reduced the concentration of ATP, they actually got 80-degree steps, 40-degree steps, 80-degree steps, and 40-degree steps. So rather than simply having 3 states, you actually have 6 states as you're rotating around. So this, as well as many other work over about 25 years, allowed groups to come up with a rotary catalytic mechanism of how they thought F1 ATPase works. And this is a little difficult to understand, this diagram, but effectively they've shown the 3 beta subunits, these are yellow subunits here, and their occupancy. So one's empty, one's bound to ADP and phosphate, one's bound to ATP. And they have a gamma subunit, this rotor in the middle here in blue. And you can see as you rotate around this ring, you can see that this gamma subunit rotates at different angles. So you have 80 degrees compared to the start, 120, 200, and so forth going around in a circle. And through a series of experiments of either putting inhibitors in, changing what concentrations they had different factors in, they could begin to dissect what should happen at each different rotary state. For example, this position here is empty and it binds ATP, so they call this the ATP binding stroke. If we then come around to a different state, you can see we've gone from an ATP to an ADP, so they see this as a catalytic dwell. They believe ATP has been hydrolyzed at this step here, and then that gets followed by this hydrolysis stroke, and they could dissect that all. But from a point of view of symmetry, you've actually got to realize that rather than having 6 steps of one subunit, because we have threefold symmetry, we actually only have 2 individual states that you ever be in. I've labeled these green and pink, and that's because you can just rotate them. So for example, a green one here is just simply a rotation, again, 120-degree rotation, because of that a threefold symmetry of the complex. So really there's only 2 states, but unfortunately structural biology was limited that it only ever gave us one of these states. So at the time I think we did this study, there were about 50 different structures of F1 ATPase in the data bank, but all of them were essentially in this catalytic dwell, although they did go either side to talk about what would happen with different catalytic inhibitor analogs. But what was missing is a structural interpretation of this other dwell, this ATP binding dwell, and that's because it dwells a very short period of time in there. And most of these experiments have been done in an equilibrium point, so an end point. So all of them were sort of stuck in this catalytic dwell. So we asked the question, how would we go about trying to understand what this particular structure looked like in order to complete this rotary mechanism scheme and show how ATP hydrolysis and synthesis occurred? So in order to do that, we utilized a temperature-sensitive mutant, which had been found along the way back in 2009, or actually a little bit before that. And in this study, what they could see is usually if you image these proteins, they almost always dwell in this catalytic dwell. So if we take this as an example at 28 degrees, we can see along the bottom here, this is a rotation angle from 0 to 360 degrees. You can see these 3 dwell states here. You can actually see some minor dwell states here, which is this binding dwell or temperature sensitive dwell, but generally it's always binding this catalytic dwell. However, if they cool this down, this particular mutant from a particular organism, that then got down to 10 degrees, the molecule spent the majority of time in this other dwell, this binding dwell, which had been unable to be seen really by structural biology up until that point. So high temperatures favor this catalytic dwell if we use this mutant, and low temperatures benefit this binding dwell. So we just modified the way we do our experiment, that if we take this F1 ATPase and introduce this mutant into it, and then we incubate it either at 28 degrees or 10 degrees in the presence of magnesium ATP, we hypothesized that we'll be able to isolate these structures using cryo-EM in either this binding dwell or this catalytic dwell. So to do that, we just took a thermocycler and we pre-incubated our protein at a particular temperature, either 28 degrees or 10 degrees, warming it up and cooling it down. And then we also pre-equilibrated our freezing robot as a vitrobot at either 28 degrees or 10 degrees. And as a control, we also did it in high phosphate concentration to understand phosphate binding. We're able to generate pretty good grids of these, although they did preferential orientation. But then when we sorted these complexes, just based in this case on heterogeneous refinement, we ended up with a system where we had these different structures, which we term the binding or temperature sensitive dwell and the catalytic dwell. And if you concentrate on these, you can see at 28 degrees, you can see about 2/3 of the complexes in this catalytic dwell. That's the dwell that we'd always seen before, which we'd had in equilibrium experiments, or that we were able to isolate a small amount of binding dwell. Whereas when we imaged it in the cold using this temperature sensitive mutant, you can see the majority of the particles were in this binding or temperature sensitive dwell. So just by changing the temperature, we could change the population of the complex we looked at. So this gave us effectively 6 structures. It really gave us two structures, a catalytic dwell, which was warm, and a binding dwell, which was cold. But you can see here, because again, that threefold symmetry in there, we were actually able to get 6 different structures of that catalytic site and be able to build in at least a moderate resolution, see how all those different nucleotides bound and the residues which were orientated in there. We were pretty confident it was the binding dwell as well, I should say, because we saw for the first time that this rotor had rotated about 40 degrees, which if you hopefully remember the single molecule, you get an 80-degree stroke and a 40-degree stroke. So we were able to identify those parts. So what was really new and novel? Well, first of all, if you compare the catalytic dwell to the crystal structure, that was almost identical. We had very similar binding, although we did see an ADP bound to an open state, which I can talk about later if you wish. But then this new dwell and the new binding dwell, as well as just having these open and closed states, we identified these new states, which we term half closed because it's halfway through closing, and half open because it's open. You can see that one of those half open states contain this ADP and phosphate, which is this catalytic dwell. So we were able to see it just post. So here we can see it in ATP, and then we're able to see it now after hydrolysis in this post-catalytic dwell. And if you look at those structures, quite obviously what's happening is you go from open, you're half closing, going to a full state. And what you're doing is just rearranging residues around that binding site, which is effectively driving either the catalysis, either way, either hydrolysis, we believe, or synthesis. So just in simple schematic, what that looks like is you'd start here in position 1 or 0 degrees. We see ATP binding weakly. That means these arginine fingers haven't engaged the phosphates here and tightly bound. We then see these arginine fingers come down onto the phosphates at close. We get tight binding. And in this case of hydrolysis, this is driving that closure. So we get an ATP hydrolysis stroke. We then get a half open state. So this is after hydrolysis, this is opening up and separating the phosphate, the gamma phosphate on the end of the ATP. And then we get open to a fully open state where we're able to get phosphate to release or ADP release through different, we can identify 2 different channels that they can release through. So what that looks like in the schematic is you have this rotation of the gamma subunit. This effectively rotates round as ATP binds. So ADP is released here. We get hydrolysis stroke, sorry. So gamma subunit rotates round. This hydrolysis occurs. So beta subunit's coming out and dragging this hydrolysis out. We call this hydrolysis stroke. And from single molecule studies, we know that's about 20 piconewton meters. And then after that, we get this ATP binding stroke. So magnesium ATP comes in and binds, and we get an ATP binding stroke, which is around about 50 piconewton meters. So this is about 80-degree rotation. But in a lab, we usually like showing videos. I don't know how well these come across Zoom, but this is looking at the F1 ATPase on the left here from the bottom. And you can see here, I've just drawn an arrow to say here is a zoom in section here of this particular ATP binding site. You can see it's empty here on the left and empty on the right. And if I start the video, the first thing that happens is ATP comes in and binds loosely here. So it's bound loosely. And as I said, these arginine fingers haven't quite engaged. They haven't engaged with that gamma phosphate on the end. And the next thing that happens is we get tight binding. So what happened there is the gamma subunit rotated around 80 degrees, and we get this ATP binding stroke. You get about 50 piconewton meters. And these arginine fingers now are coming and tightly binding down on that ATP to drive that rotation, or obviously in the other direction, we're talking synthesis. Then the gamma subunit rotates around this closed state like a pivot as it comes around the side. But we're essentially in the same conformation in the catalytic dwell, just pre to catalysis. And then as soon as the catalysis happens, this opens up, those arginine fingers move outwards, and we get that 40-degree rotation. And that stabilizes this separated state where we have phosphate, inorganic phosphate by itself, and ADP. This then opens up to release 2 channels. The phosphate's able to release through a backdoor channel, the ADP releases, and this just cycles around. And this is how we're able to, in this case, generate rotation or generate torque by simply binding and having a hydrolysis event as a gate to cause that rotation. So in summary, this F1 ATPase has 2 strokes, an 80-degree ATP binding and a 40-degree ATP hydrolysis. We've now seen it both by single molecule and also provided a structural representation of how that's happening. And these arginine fingers are crucial. They both involved in binding, ATP binding to cause that closure, and also catalysis to open up and separate ATP and phosphate. We also identify a backdoor that's able to allow independent binding of magnesium ADP and also phosphate, although we're still unsure how these binding sites communicate with one another, which is an active part of our research. So hopefully that's a bit of an overview of some of the work we've done on F1 ATPase. And I think I really like that study because it also shows how we're able to kind of come up with an idea of answering a specific question by using a specific mutant. But the next piece of work we did was look at the whole F1Fo ATP synthase, which I'd done beforehand. And the reason why we did this is there were a whole series of mutagenesis and studies that had been done, particularly in this FO region. And we hope by providing a structure, we could begin to really understand a molecular basis, at least of how bacteria ATP synthases are able to function. So in order to express the protein, we utilized a method which was developed by one of our collaborators, Robert. And here we just taken E. coli and ATP synthase, and they've knocked out all of the genes of ATP synthase, luckily all in one operon, so it was relatively easy. So we call this a DK8 strain, but it's just a knockout strain, so it has no ATP synthase. We can then take a plasmid, which has that whole operon on, and introduce it back in the bacteria, and introduce a his tag on one of those beta subunits. And we just took their purification, or I should say Meghna did, but she spent a lot of time in optimizing it really for what turned out to be cryo-EM at the time. She did so by changing the purification conditions to move over to digitonin, and that's because she found that she could have a much better gel filtration trace, but also the complex lasted a lot better. So previously in these studies, when they'd used quite a large mix of different detergents and other factors, they found, if you look at the circles, that over time, the ATPase activity reduced drastically. But if we change this to digitonin and purify it using gel filtration, not only do we get monodispersed protein, but it lasted a lot longer just on the bench or in the fridge. So we could be quite more confident we could use it for these structural studies. So Meghna performed structural studies on this to make our lives a little easier. We incubated with 10 millimolar magnesium ADP, and that's because we hypothesized that it would stabilize the complex in something called the ADP inhibited form, and that should make it hopefully nice and ordered and structured. And when we did this, we got what I thought were beautiful 2Ds. You can see these peripheral stalks on the side, even individual helices in these 2Ds. And we also got very nice orientation of those particles. And when we came to solve the structures, it was pretty nice. We went down to about 3 angstrom. But what happened is in this F1 region, which is what I spoke about in the first part of this talk, we had lots of high resolution features. But in this FO region, which was probably the region we were most interested in, you can see there's hardly any information. And I'm sure most people on the, and you can see there, we also got it, sorry, in 3 states. Hopefully you can see here the rotor is in 3 different rotary states. We had these 3 different rotary states. As I've shown you, there's 120 degrees. But because it was locked with ADP and also in an equilibrium state, we would have seen that single effectively catalytic ADP inhibited state. But what we went on to look at is, well, if this area has pretty poor resolution, it's probably blurred out due to the factors in multiple different orientations. So this was performed in Roland 3 at the time, where we've got a mask. We masked out this region, which was pretty poor density, and just did, or we relied on this F1 and then did 3D classification based on this FO region without image shift. And hopefully you can see there's a whole series of structures now, which although they're in the same F1 conformation, this stalk on the outside and the rotor is in different rotary conditions. So this piece is effectively rotating relative to the F1, which we believe is why it was blurred out in those structures. And that allowed us to get a whole series of different states at the time based on these. So you have rotary state 1, 2, and 3, and then within that we have all these different substructures, which are showing the molecule in slightly different positions relative to one another, and allowed us to build effectively a molecular model of this FO region to begin to understand it. So the first thing we looked at is the enzyme really inhibited? There's this inhibitory subunit, which is I've shown here in green, which is called subunit epsilon, been hypothesized beforehand by Tom Duncan and also Mitchell Gus over at University of Sydney, that this might be able to transition between what we call an upstate and a downstate. And largely because we imaged this in magnesium ADP, you can see this in an upstate, confirming that this was certainly possible in a F1Fo context, which was still not widely believed in the field at the time. But as well as this now, because we can build atomic models down the bottom here, we can begin to understand this whole myriad of mutagenesis work that have been done here in Canberra down at ANU by Graham Cox, was started by Graham Cox and Frank Gibson, where they began through and just started mutating out different residues, particularly in this orange and gray subunits, to understand how, I believe, what were the important residues, and potentially trying to understand the function of how this worked. So I think the most interesting two mutants, which rather than going all over them, I'll just talk about two mutants. The first mutant was when they removed this position 210, they got completely devoid of any ATPase activity or a hydrolysis or synthesis activity, actually. So by doing that, they realized that this position arginine 210 must be very essential for how this molecule works. More than that, they realized if they took this position R10, R210, and they also Q252, and they could swap these two residues, they could actually regain function. So they could, by swapping these mutations round, they could regain function. So they must be located near one another. Now, at the time, they thought these residues were vertical, effectively, in the membrane, or perpendicular to the membrane. Whereas we know now from other people's studies before ours, and also ours now, that we have these helices, which they originally thought were perpendicular or are sort of inclined or almost parallel to the membrane. And you can see here this arginine 210 is indeed very much located next to this Q252. So this arginine 210 is essential, and you need to have it in this particular location here. We could also see these two different proton channels, one on the cytoplasm and one on the periplasm. So protons are able to come in through these channels and bind to some sort of ring here, but they're prevented to getting through all the way through the other channel by this arginine 210 by having this position here. And if you rotate this round and look at it on the other side, as well as understanding what other people hypothesized before, you can see that arginine 210 is interacting, in this case, with this aspartate in order to neutralize this aspartate so it doesn't need to bind a proton. So protons come through the periplasm, they bind to this acidic residue, they have to go through a full rotation around the outside, and then they're released into the cytoplasm on the other side and bacteria. And you can see how this arginine is able to effectively take that proton off, and then you're able to bind to the arginine so you don't get any short circuiting. So for example, you can't just get the protons going in another direction, or they might go all the way around. And this gives now a molecular interpretation of how this F1Fo motor functions. So as well as that, understanding, at least now we've got good maps, we also were left the position that we actually had all these different states or all these different substates of what this molecule looked like. So what could they potentially be? So again, we used a movie morph to understand what these different substates were. I'm just looking at state 1, and you can see we had 5 different states that came up from the classification. We had A, B, C, D, and E. And if you look at this green dot here, it moves backwards and forwards between the A and B, and that gives you an idea of taking that interpolation between, or morph between, the A and B. And if you look between the A and B, you can see that we can actually see rotation of 36 degrees of this rotor in the middle. So we're seeing actual physical substepping of that Fo subunit. But if we morph between B, C, D, and E, you can see actually we're looking as just torsional movements of this molecule almost acting like a spring, so moving backwards and forwards. We don't get rotation, we're just getting movements of this F1Fo complex. So again, coming back to here, what we saw is we saw this Fo substep going from state 1A or 1 prime, we call it. And then all these B, C, D, and E, they're effectively the same rotary state, but we're just seeing flexing of that object. And then over in state 2 and 3, these rotary states, we don't see any substepping, we just see this rotary state. And why would that happen? Well, it makes sense if you look from the bottom and look at this rotor. So at the top, as I said, we added ADP, so it's locked in these 3 inhibited states. But at the bottom, this Fo, this proton ring, where we have these protons binding to the ring and causing rotation, we have 10 of these. And because you can't divide 10 by 3, what's happening is you get 3 of these rotations, you get 3, and then you get 1. And that's how you're adding up to 360 degrees. So rather than having, I guess, 3 rotary states in the Fo, we get 4 rotary states, a state 1 prime and a state 1 double prime going around. Now, we thought this was pretty interesting. I should say, preface this with this didn't make it through review. I think I agree with the reviewers. There's a lot of error involved in this. But one thing that Richard realized that we could work out here is if we have a system where we have relative number of particle numbers, if we believe those particle numbers are true, we could actually work out the elastic spring constant of our molecule. Now, it's probably only really related to ATP synthase here, but again, what you have is at the bottom here, these F1 dwellers are separated by 120 degrees. You have state 2, 1, and 3 arranged in a line. Here we have this C-ring dwell, so this is the proton binding dwell. We have 10 of these, so you can count from 0 out to 5 and 0 out to 5. And if we believe it's a linear spring or a molecule is acting like a linear spring, we could have a parabola of energy on a particular chart. And you can imagine that in these positions here, as I've shown you on the previous diagram, where you have the F1 ATPase, its dwell in the F1 ATPase is coincident with an FO motor, or this C-ring, we'd be able to see that. So I've drawn these as a dark square, so we can see this state. But this state over here, which is too far away from those F1 dwells for the spring, we don't observe it in this equilibrium experiment we have again. So I've drawn that as an empty square. Whereas you can imagine here at state 1, as I've already shown you, we have state 1' and state double prime. And that's because this F1 dweller is somewhat in between these two dwells. So that means we're allowed to see this on this energy diagram. And then finally with 3 again, because it's coincident with an F1, we only see one of those dwells. So if we rearrange Boltzmann's law, we're able to work out KT being around, because we just look at the relative numbers of particles we find in state 1' and state 1 double prime, and we get a KT of about 16.4, which works out to about 40 kilojoules. And the reason why we thought this was quite interesting is it's comparable to the delta G of ATP, if you remember at the start of the talk. So by looking just at the particle numbers, we're able to derive a spring constant for the molecule, which is almost perfectly matched to ATP, which is what you'd expect, or hopefully you'd expect in a molecule. But again, as the reviewer said, I suppose with these particle picking programs, I suppose it's not an exact science. We don't know if we get bleaching of these particles from one particle to the other, but it was still quite a pretty experiment. So in summary, magnesium ADP addition results in this inhibited E. coli enzyme. We've shown that E. coli synthase contains 10 of these C subunits, these proton binding subunits. We defined the substepping of the FO motor, showing how this flexible molecule that has some sort of spring constant can mediate this rotation of 3331, or 3331, at least in this catalytic dwell. And we provided a structural model of bacterial FO rotary mechanism, complementing that mutagenesis study that had been ongoing through the 80s and 90s. So hopefully I've got about 10 minutes left. So I'll really try and brushly go over another subunit, or another protein that's worked in my lab, which was really driven by a very talented PhD student who's just written up now, Jack Zheng. He's in his third year of his PhD. And what he wanted to do was try and understand how these human organic cation transporters work, which is a family member of the SLC22. So these are organic cation transporter 1. So this is part of the SLC22 family of organic cation and anion transporters. Obviously, the cation is part of those. And the MFS drug transporters with broad substrate and inhibitory specificity. So there's well over 100 different drugs or substrates known that are either transported or bind and inhibit this particular transporter. As a result, it has roles in drug disposition and drug-drug interactions. It's enriched in the liver, so it's often involved in drug metabolism and hepatic clearance. So reduced function variants of this particular molecule, as well as drug inhibition by ones that bind to it, shown to alter efficacy and tolerance to substrates, particularly as the frontline drug well known as metformin, but also other cardiovascular drugs. So this is a MFS transporter, which you might have heard before if you're in a sort of transporter field. But for those that haven't, it's the major facilitator superfamily. And there's evolutionarily conserved by this 6x6 transmembrane fold. So if you look on the right here, you can see these 2 bundles of transmembrane helices and a 6-6 giving you these 12 transmembrane helices. They're usually involved in passive transport, and they are believed to always operate by this alternating access mechanism. So you can see here you have these are these 2 different bundles just shown as ovals, and you'd have an outward open conformation. Substrates would come in and bind to some binding site in the middle here, and then they would go through an occluded state and come to an inward open state and then be able to be released on the other side of the membrane. And these generally show high substrate specificity. People probably have heard of the GLUT transporters. But in the case of these SLC22s, instead, they seem to have very broad substrate specificity and be able to bind a whole series of drugs and substrates. And again, just with the ATP synthase work, we were a whole group of different teams which published structures of these around a similar time over the last year or two. And other people have been working on, for example, OCT3 or OCT1, and these have been shown in the outward open structure. But I guess where our work became unique and really completed that story is we're able to see this human OCT1 in an inward open conformation. So hopefully you can see from the maps here, these are sort of pointing outwards, as I showed in the axis mechanism, whereas this is pointing in an inward conformation. So how do we express this? We took advantage of the Bachmann transduction system with HEC293 cells, and we also utilized this GNTI cell line. And that's because preliminary data had shown that we've got a few species with complex glycosylation. So by removing the complex glycosylation with this type of cells, we're able to just get a single band. Solubilize this in either or a few different detergents, used a nickel purification again, and size exclusion probatometry was also cutting that GFP in order to get a single band. Could then take a few movies, extract a few million particles, do 2D classification and 3D classification. I suppose a bit like always a standard protocol. And we're able to get around about 3 angstrom structures of this OCT1 protein. So Jack was very talented. He was able to solve 2 structures. And I thought this was pretty interesting because all that's really changed between these 2 different structures is either he's purifying the protein and imaging it in GDN, or he's purifying the protein and imaging it in LMNG, a mixture of LMNG and CHS. When he purifies it in GDN with different substrates, he's able to see this intracellular helix and intracellular helical bundle and extracellular domain up the top. And it's in this outward conformation, outward open conformation. But when he images it in LMNG and CHS, he sees it in an inward conformation and we're unable to see this intracellular helix potentially because they're moving around. So by morphing between those 2 states, we can begin again to understand this full alternating axis mechanism. So again, apologies if this doesn't come over Zoom, but this is showing a morph between those 2 conformations. You're seeing the inward conformation, inward open conformation now, and now you're seeing the open outward conformation. And by oscillating between those through an occluded state, you could take substrates from one side of the membrane to the other or backwards. And what was different about this structure compared to what had been seen before, because now we can see it in this inward state, is usually these gates on the extracellular side are made up of hydrophilic interactions. But in this case, we saw this hydrophobic gate. So this kind of gate, which is all made of these hydrophobic residues you can see on the right here. And we think this might explain why we have incredibly high fast kinetics of this transporter compared to some of the other previously well-known MFS transporters. So Jack also took a series of different substrates or inhibitors, looking for a range of different things. You can see here he chose these not only because they had different scaffolds. This is a substrate believed to be an endogenous substrate, thiamine, but also quite well-known drugs like metformin, a bronchodilator, phenetrol, or diltiazem, antihytensive. And he also chose these because they had different scaffold, but also because, for example, here with diltiazem, you have these point charges, or you can have this diffuse cationic charge across the molecule. To begin with, he looked at using an inhibitory transport assay. So here you're looking at fluorescent compounds. You expose your cells to them, and then you put increasing concentrations of your substrate. So you effectively can work out IC50s and look at inhibition of transport of that fluorescent compound, of what doesn't get inside the cell. But then a much better assay that he developed afterwards was an LC-MS-based assay. So this is a mass spectrometry-based assay where you're washing these substrates or inhibitors onto the cells and then seeing what goes inside the cell by quantifying them afterwards with mass spectrometry. And you can see, for example, with thiamine metformin, you get quite strong uptake. Looking at the y-axis here, you get very strong uptake of metformin. Phenetrol, you get quite weak uptake, but with diltiazem, he was able to confirm that it is inhibitor. So versus control, we're not expressing OCT1. You can see there's no difference between expressing OCT1 or not inside these cells, which was sort of what unknown at the time. So you have these tightly binding substrates, which either are very weakly transported or not transported at all. And then you get these weakly binding substrates, which have very high transport efficiency. So he was able to solve structures with a few different substrates inhibitors, but I'll just talk about 2 of these in the interest of time today. So both metformin and diltiazem. So with metformin, he's got structures in an outward open state and also an inward open state, and you can see where they bind in the inside and define that binding pocket. Quite strangely, it was predicted to bind over E386, which you can see on the right here, which is up here on these diagrams. But in fact, he sees it more centered inside in this more diffuse sort of cationic binding site by this probably diffuse cationic charge around this molecule. He's able to mutate out the different regions and see that pretty much if you mutate any of the key residues around here, you get reduced function of transport inside his transport assay. Ada Quinn and Megan Ma did some beautiful molecular dynamics on this system. And not only can see where it would bind inside the binding site to confirm the resolution, given it was only about 3 to 3.5 angstrom, but they're also able to even see an event where they can see metformin being released in the open state, which we thought was pretty exciting. So now diltiazem, which is a slightly different molecule, this is much larger than metformin, and also has this point amine group on the end here. And this point amine group, we believe this whole structure by having this hydrophobic region, but also this point amine group is binding to that E386 region and preventing the progression and release of this particular molecule as an inhibitor. So by seeing it in the binding site there, it's potentially because it's large, but also because of this point amine group pointing towards and binding with that E386 right on the edge of the binding site. So by combining all this data together, Jack was able to come up with an interpretation of this alternating access mechanism of how you'd go from an outward open state where you have this outward facing conformation and you have these intracellular helices on the inside also providing part of the gate. You go through an occluded state where we haven't seen yet, at least in our study, and then you go to an inward open state where this intracellular helices move out. Actually, cellular helices do move across, extracellular domain move across, and then you get release of your substrate on the inside. So if you take a very large molecule with a point charge, something like an imatinib, an anti-cancer therapy, this would be so large that it doesn't allow the extracellular gate to shut and shut close. Whereas you take a small substrate, such as metformin, this is able to bind in an outward open state, come down to bind in this region here. You then get closing of this gate. I believe there's a change here where we get charge neutralization of the arginine with this E386. And this begins this intracellular gate open, extracellular helices open, the TM's open, and you get release of metformin on the other side. Whereas a molecule like diltiazem seems to be able to go through all the way to the inward open state, but it seems to get prevented from release by this point charge still binding to that E386 on the side of the binding site. So in summary, we defined the alternating axis mechanism of this human OCT1 and suggested how a unique hydrophobic gate may dictate rapid transport. We've shown how different drugs are able to bind to OCT1 by combining both functional and structural data, and shown how we believe these large point charges are able to bind to the transporter and inhibit transport, hopefully providing a framework to understand these drug-drug interactions. And finally, we've also now got inward and outward structures of metformin, and we'll combine this with mutagenesis and molecular dynamics to show these key residues which are involved in the transport or full transport cycle and metformin. We've got about 10 minutes left. And I've already thanked everyone involved, but particularly Jack Zhang for driving the OCT1 work and Magnus Opti for driving the ATP synthase work. And thank you. I'd be happy to take any questions.
Thank you so much, Alastair, for the amazing talk. And now the room is open for questions. Okay, we have a question from Theodore. Please unmute.
Hi, Theo.
Hey, Alastair, how you doing?
Good.
I had a question about the extracellular and intracellular domains of this transporter. I was wondering what the function of those are and how stable they are relative to the TMD.
So I kind of vaguely said that, but I guess Jack is able to see these extracellular loop very well in the inward conformation, whereas it's generally moving about a fair bit in the outward, which is can't really see it too well. In the intracellular helix, in other transporters like Lutz, it's been pretty well shown that this is part of the gate, as in it would come round and almost block part of the gate. We don't quite see that in these structures, but it's certainly synergistic that it would be, partly controlled by the gate. In terms of their function, certainly intracellular helices are probably part of the gate and part of that work. I think, interestingly, I didn't go into that, but actually how these SLC22s is really classified, as well as being broadly diverse, is also this extracellular domain, the piece on the top. Yeah. We expected maybe to find some sort of function for it, but the only thing Jack has been able to see is certainly if he mutates out the cysteine residues in this extracellular domain, it prevents trafficking as it doesn't really get to the cellular membrane. Now, so it's potentially to do with stabilization. It certainly has glycosylation on it. We can see a few different glycosylations on it, but in terms of like function of the protein, we certainly haven't seen anything to kind of show that. Did that answer your question a little bit?
Yeah, definitely.
Yeah,
Thanks.
okay.
Do we have more questions?
Brett, I think.
Yeah.
Yep.
Brett.
Oh,
Hi, Alastair. How are you?
good. How you doing?
Good. Yeah, beautiful talk. It's probably related to both parts of the talk, actually, but maybe starting with the transporters. So you see different conformations with the different detergents that you're using to extract it. Do you see any indication that your different conformations are stabilized by potentially a bound lipid at some particular site, or?
Maybe. We don't really see them in a particular site that would put them together. Again, I think because these are around about 3.5 angstrom, we struggle to see like clear lipid densities. With the ATP synthase work, where we've gone a fair bit higher resolution, is generally when we start below 3 angstrom is when we've really started being able to identify lipids. I guess my feeling is you're certainly changing the environment. One of them, for example, Jack, added cholesterol, for example, so it's going to make it much more rigid and move. I should say on published work as well, but I didn't put into this talk. He did solve it inside nanodiscs, and that just became far too complicated to be able to solve structures of. So he could never really get below sort of helical resolution. And our interpretation of that is, at least in nanodiscs, you were getting everything, as in everything was moving around, that it just became too challenging to sort in any way. So I can't tell you exactly what's happening. I guess certainly in this outward open, you get, maybe the diagram even shows it, there's a bit of a fenestration. It actually opens up to show the membrane in a way. So you'd imagine that potentially, in this case, GDN could be coming in and binding and kind of preventing closure there, or at least preferentially moving towards this outward open state.
And with the ATPase as well, then, do you see any, you mentioned you can start to see some lipid densities. Is there any suggestion that there are lipids that are regulating different conformational states or anything?
Yeah, so when I took that slide out, but effectively Carol's kind of done a fair bit of work in that area and certainly looked at our preparations. So we generally take these molecules obviously from their native organism, E. coli. And if you perform lipidomics on E. coli versus our preparations, which are purified just with digitonin, you can see a huge enrichment with cardiolipin, which has been shown with many other ATP synthase as well. So we hypothesize that the lipids we see are cardiolipin, but again, around about 3 angstrom, we can't fully say what they are. But at least in our structures, we also, well, I don't know if I can show you quickly, probably not, but we see a bridge between those C rings and the A subunit. So you can imagine you somehow have to have a protein-protein interaction, which is both specific and holds the thing together, but then also allows this huge conformational change or rotation between it. And we see those lipids at that interface in between. So we think that might be a way that you can sort of have this semi kind of specific interaction rather than having protein-protein, but having sort of protein-lipid, lipid-protein interaction between them.
Yeah, cool.
Yeah, thanks.
We might take one more question. Okay, I have a question. Okay,
Oh, yeah, sure.
For the cryo-EM structure, that for the ATP synthase. Okay,
Yep.
You mentioned that you used the digitonin for grid preparation and solubilization of the membrane protein. So was it like below CMC or above CMC? Because it's
It was
it's a bit tricky.
that prep was basically like on CMC, I think.
Okay.
So generally with the LMNG, for example, we just put it without any detergent over the size exclusion column.
Okay.
It's a bit tricky to say though. So in that study, we're using digitonin, which I believe in most people's hands seems to crash out in cold rooms or like in 4 degrees. So generally what Megan likes to do is make digitonin up the day beforehand at like in this case 0.005 I think percent it is, and then she puts it in the cold room, and then quite a bit of detergent generally crashes out of solution, or we get white precipitate, and she sterilized filters that to remove that precipitate and then uses those buffers. So I can't tell you the exact concentration of digitonin in the buffers because we're effectively losing some. But when we do that, we seem to get nice preps and we get nice purifications of our protein. And
Okay.
that seems to be, across the board with other molecules we worked on, that by preparing digitonin in such a way, it seems to be happy on the column, whereas if we put no detergent in there, it isn't. But it seems to give it a minimal amount to be happy and move forward, and not, obviously crash out on the column, which can be quite bad.
Mhm, mhm.
Want to do that.
Okay. Any more questions? So, okay, with this way we will close our seminar. And again, thank you so much, Alastair, for today's amazing seminar. And thanks for all audience who attended the seminar today. Please remember that the date for upcoming seminar will be held on June 11th. And our guest speaker, Dr. Carol White from Dimex Company. Enjoy the rest of the day, everyone, and bye-bye.
Thank you.
Thank you so much. Bye-bye.